Great. Thank you to the organizers for asking me to step in. I appreciate the opportunity. I want to start off today by focusing on some of the a summary of some of the basic science that has led us to our design and rationale behind some of the novel small molecules that we've developed that we think will be effective for peripheral neuromuscular disorders. And this revolves around some work that I won't show some of the data for, but I just want to highlight our motivation. So this work has all been done in the mouse neuromuscular junction, which is a very large synapse with many release sites for transmitter. And here in this image, the red stain is a postsynaptic acetylcholine receptor stain. And the green is antibody against bassoon, which identifies the presynaptic active zones or transmitter release sites. Within any one of these green spots, if you reconstruct the EM tomography data, you find that there are double rows of ion channels, including the CAV2.1 or PQ type channel. And between those double rows of channels, there are 2 or 3 synaptic vesicles that are docked, ready to be released. And despite the fact that you have 700 of these active zones in a single motor nerve terminal on one muscle cell, each with 2 or 3 synaptic vesicles ready to go, when you stimulate the axon leading into this synapse, you end up only releasing about 150 vesicles. This derived from intracellular recordings where you can measure the postsynaptic end plate potential as compared to the miniature synaptic potentials that indicate the release of single vesicles. So really, this synapse is strong and reliable. And every time we go to move our muscles, it works because we release more than enough transmitter than needed to bring that postsynaptic muscle cell to threshold. And we can do that repeatedly because we're really only using a subset of available resources. And so each individual active zone is really not participating very often. And one of the early basic science questions we addressed, and I won't go through the data, but I'll just summarize the results, was what leads to constructing individual transmitter release sites as low probability release sites? Number one, there are not very many calcium channels, we don't believe, in the active zone. And these don't open with a very high probability during a natural action potential. We've made voltage-sensitive dye recordings recently of the action potential in these nerve terminals, and it's very brief, on the order of 300 microseconds duration at half amplitude. And so you're not opening very many of these calcium channels. In fact, we predict that in some active zones, you would randomly not even have a calcium channel open during a single action potential. So that's one, you're not opening calcium channels with very high probability. A second low probability event is that even when you do open a calcium channel, the triggering of vesicle fusion by the flux through that one channel under normal conditions is not a high probability event either. And so you're really not releasing transmitter all the time, even when you open a calcium channel in the active zone. And so, in thinking about how the synapse normally works, we kind of apply these lessons to what happens in disease states. And one of the diseases I want to talk about today is LambertEaton myasthenic syndrome. This is a rare, about 1 in 100,000 people, autoimmune disorder in which patients get a peripheral muscle weakness. And it's an autoimmune disease that targets the presynaptic calcium channels. So here, this reduction in calcium entry essentially decreases the probability for release at these synapses. So you have antibodies that the patient's making, and this can be as a result of really 2 situations. One, about half of the Lambert-Eaton syndrome patients, this is paraneoplastic, so they have small cell lung cancer, and this is a neuroendocrine tumor that is believed to trigger an autoimmune attack of a lot of presynaptic proteins, predominantly calcium channels. Or 2, they have no history of heavy smoking or cancer, but for some reason they just develop autoimmune disease that attacks the same proteins. Either way, they have the same disease, and these antibodies are thought to cross-link and internalize the presynaptic calcium channels. And from freeze fracture studies, the interpretation is that these active zones break up so that the presynaptic double rows of ion channels are now disrupted. There are fewer particles in the freeze fracture, and the extracellular recordings of calcium flux suggest that there's a reduction in the total number of calcium channels present. This, of course, reduces transmitter output so that now in Lambert-Eaton syndrome patient, there's insufficient transmitter release to bring these muscle cells to threshold, and these patients are weak, they're unable to get out of bed, or they're wheelchair bound, so it's very devastating for them. So the strategy has been, because the immunotherapies have too many side effects, the strategy has been a symptomatic one. Try to increase the calcium entry from the channels that you have left. You know, this is a balance between the immune system attacking and removing channels and the motor neuron overexpressing channels. And so these patients don't progress significantly after the disease is fully manifest, but rather for the rest of their lives, they'll have this kind of balanced weakness that results from their motor neurons constantly overexpressing to try to keep up. Fortunately for us, in trying to use mouse models to evaluate potential therapeutics, calcium-triggered transmitter release is one of the most conserved mechanisms and is expected to translate well to humans. So what's the current therapy? Recently there was FDA approval for potassium channel blocker, 3,4-diaminopyridine. We've heard about potassium channels a lot in this meeting, and DAP will broaden the presynaptic action potential, basically increasing the probability that those calcium channels will open. And so you'll get a larger calcium flux, but there are dose-limiting side effects. You can't broaden your action potential too far, or you have problems, and so these patients really only have modest recovery as a result of the clinical doses of DAP, which are really only thought to bring the plasma DAP concentration in the 1 micromolar range, so really not very high. So we were motivated by a different strategy. DAP solved one of the 2 low probability events that we uncovered in our basic science studies, the low probability that calcium channels would open, but we wanted to solve the second low probability event, which is that calcium flux would be sufficient to significantly trigger transmitter release. So we started working with roscovatine. This is a cyclin-dependent kinase inhibitor that is in clinical trials for various forms of cancer. And Gringard's group showed that there was a significant side effect, if you will, of this CDK inhibitor that was prolonging the deactivation of the calcium current. So here, with an action potential or a long square depolarization, you can see that channels that normally deactivate quickly now have very slow deactivation. And at the single channel level, single calcium channel openings that are really very brief normally, in the presence of roscovatine, you can see prolonged openings so that it seems to stabilize the open state of the channel. The only problem, of course, is roscovatine is a cyclin-dependent kinase inhibitor, and we were not interested in using that moving forward. So we knew, based on published crystal structure work of how roscovatine bound to the cyclin-dependent kinase, that there were side groups that we could target and tried to change to eliminate the CDK activity or greatly reduce it. So I went to the chemistry department and talked to Peter Whiff, who's been a very effective collaborator on this project. And initially we made about 20 or 30 such changes to those side groups, finding that isopropyl to N-propyl changes would significantly shift the CDK2 activity on this side group, and that changing from benzene to a thiophene would also accidentally, because we knew nothing about how this molecule would bind to a calcium channel, would actually increase the deactivation slowing that we detect with roscovatine. So in these plots, I'm just showing kind of the end of a long depolarizing step. So this is the repolarization. And normally in control currents, they repolarize quickly. Roscovatine repolarizes more slowly. And some of these analogs, particularly this one in blue, repolarizes even more slowly. So these are prolonging the mean open time of calcium channels. And we focused on that one molecule that we now call GV58, which has 2 side group changes, and significantly slows the activation of the calcium channel in a manner even better than roscovatine. And furthermore, that shift in CDK activity is relevant physiologically, because if you just look at cell proliferation in culture under normal physiological ATP levels, GV58 does not inhibit proliferation, especially not near the EC50 for this activity on deactivation. But roscovatine, you can see, will inhibit proliferation at doses that it also acts on calcium channels. So our novel analogs, and now we've made over 200 of these now to try to optimize this. Some of them have 20-fold lower affinity for CDK, 5 to 6 times higher for the CAV2 channels. And these are CAV2 selective, so they do not act on CAV1 or CAV3 channels, and 4 times stronger potency. One of the important properties of these molecules is that they're voltage-dependent action. So in most of this work I've shown so far, we're giving long 100-millisecond steps, which is long enough to open the channels repeatedly such that most of the channels are modified. But if you give very brief depolarizations, as you might expect during an action potential, only a small subset of the channels are actually have the opportunity to be modified. So there's a kind of an on rate for modification for this molecule. And the longer the depolarization, the greater the number of channels that become modified. So we kind of speculate that this molecule is only modifying channels when they're in the open state, and that if you only briefly depolarize the cell, then there's not enough time for the small molecule to modify all of the channels, only those that it can get to. Kind of another interesting observation was that the magnitude of modification depended on when you applied the modifier. So here we're using a different analog of roscovatine, but it has similar actions as GV58. And we're applying it using a fast perfusion system where we can change the solution around the cell within about 25 milliseconds. And we can either apply it during a long voltage step, so these are 200 millisecond voltage steps, and then continue through the tail, or we can apply it before we depolarize. And what we find is if we apply it during the voltage step and then continuing after, the effect is very limited, as compared with the effect in the dotted line here that is present when you apply it throughout the experiment. On the other hand, if we only apply it for 200 milliseconds before we depolarize and then wash it off for 200 milliseconds, this brief application during the closed state of the channel is enough to give us very strong modification of the current. So we interpret these to suggest that the compounds gain access to the channel when they're in the closed state. So maybe they only modify the channel in the open state, but they can only really effectively gain access in the closed state. And so our model looks something like this. We think that GV58 and its related analogs, which are uncharged at neutral pH, partition into the plasma membrane, from which we think gain access to the channel through a state-dependent fenestration in the side of the channel, much like has been described for sodium channels. And that once inside the fenestration, if the channel gates to the open state, a binding site is exposed that the compounds can then bind to and then stabilize that open state. In fact, we've made some homology models. There's no structure of this AV2.1 channel. We've made homology models based on the sodium channel open and closed states that have been published for this PQ-type channel. And what we find is that there is, in the closed state, a predicted fenestration that is pinched off in the open state by 3-phenylalanines around the neck. And we're in the process of evaluating that. Furthermore, we've made homology models and evaluated the effects of different regions of the predicted structure, and then tested those predictions with mutations. And so, for example, the CAV1 channels are not modified by any of these small molecules, and therefore you see no slowing of deactivation. But if you make a chimeric channel, in which most of it is CAV1 in blue, but the domain 3 is CAV2, then you see modification. And furthermore, this is consistent with what was shown for roscovatine, is similarly sensitive to domain 3 being CAV2. So if we use AutoDoc to try to look at predicted binding sites in our homology models for open or closed state channels, and then focus only on those sites that show a binding region in the open state where we think it's acting, but not in the closed state, that exist within domain 3. And then furthermore, we've narrowed that down so that we know if we make a chimeric channel that contains segments 1 through 3 of domain 3, it's not enough to impart sensitivity, but 4 through 6 is. So we demand that it be in the region of the channel where we have segments 4 through 6 of domain 3. And we find that some of those amino acids within that region, which is circled here, that is binding in the open state but not in the closed state, if we make mutations of some of those amino acids, here we're mutating the amino acids for which there is a different amino acid present in CAV2.1 versus CAV1 channels. So we're swapping the CAV1 residue for the CAV2.1 residue. And you can find that some of these mutations greatly reduce the time constant of deactivation here. And especially if you combine 5 mutations together, then you get a significant reduction in the slowing that occurs. And so based on these data, and we have not narrowed this down, it's still in progress, but we're speculating that there's a binding site somewhere in this region that includes segments 5 for domain 3 and domain 4 at the intersection of those two domains. But given this effect, then the question is, what about the potential for therapeutic benefit? And we've made a mouse model of LambertEaton syndrome. You make this in the lab by collecting blood from LambertEaton syndrome patients that includes the antibodies directed against calcium channels and other presynaptic proteins. And then if you just inject that serum once daily into the mouse for 16 to 30 days, that creates kind of a passive transfer, and the animal develops the same kind of weakness that the patients have. And so in order to test this concept, we've actually decided to combine the 3,4-diamidopyridine that the patients already take, which prolongs the action potential and solves the first low probability event, increasing the number of channels that open, with our compounds which solve the second low probability event at neuromuscular synapses, which is the calcium flux is normally not sufficient to create a high probability for fusion. We can evaluate these two, predicting a synergistic effect between the two based on their mechanisms of action. And if you measure the end plate potentials from mice that were either injected with control patient serum or LambertEaton syndrome patient serum, you can see that the postsynaptic end plate potential gets tremendously reduced in those mice that receive 16 to 30 days of LambertEaton syndrome. Our compound by itself will double the amount of transmitter release, and that's similar to the effects of 3,4-diamidopyridine by itself that the patients currently take and only receive modest benefit. But if you combine the two, you can get complete recovery of the end plate potential size postsynaptically. So we think that this has the potential to be a more effective approach to dealing with this presynaptic weakness that exists at the neuromuscular junction. We've started some behavioral studies on these as well. So we use grip strength to assess muscle strength and IP injections of these compounds, and we've seen a recovery that's preliminary, so I don't have the data yet, in function in vivo as well. But we have done more work with another disease, and that's spinal muscular atrophy. So currently this disorder is in the news because of some recent FDA approvals for gene therapy to replace the SMN defect, and that does keep the motor neurons alive, but there's patients still have neuromuscular weakness. And so this is projected to be a potential add-on therapy to increase strength in these patients. And in fact, You can increase transmitter release with GV alone or in combination with DAP, and in grip strength measurements of these postnatal day mice, you can find that you also get recovery in strength. So I think this approach we've shown now is effective in a variety of conditions, and we predict that it might be effective in even more, and essentially any condition in which there's a presynaptic source of the weakness, this approach will work. I didn't say, but it's important to note that these compounds are not blood-brain penetrant very well, so they stay in the periphery. That's why we're talking about neuromuscular diseases, and further, I think they would potentially cause problems in CNS, perhaps seizures. So with that, I want to acknowledge the graduate students and undergraduates in the lab who've done all this work, our chemistry collaborators who've synthesized all these compounds, our clinicians who provide access to the patients, and of course, mostly the LEMS patients who've decided to participate in these studies and donate blood, and our support. Thank you very much.
Questions? Yes.
So can you actually rule out that CDK phosphorylates the channel? What if the channel is a substrate for CDK? Because the drug that you're using don't inhibit CDK,
Right.
don't affect proliferation, but what if they stimulate CDK?
So, Russ, it's been examined in Roscovatine, where Keith Elmsley's group has shown that these effects are CDK independent. And so those experiments have been done for Roscovatine. We haven't repeated them for our analogs, but we would expect similar results. Bruce.
How long does the modification of the channel last, and will it build up with some frequency of firing of the presynaptic action potentials?
So we haven't done trains of action potentials to look at this yet, although in patch clamp studies, primarily because expressed 2.1 channels for us require holding potential of -100, just because there's a shifted steady state inactivation, and I'm still a little bit not sure about how to apply an action potential waveform from -100 and predict that it's reproducible, but we could do that experiment. We just haven't. The modification typically, we think, prevents the channel from closing depending on its off rate in that fenestration, but this is just a hypothesis. We have yet to test that.
Have you thought about looking at any of the compounds in the context of the familial hemiplegic migraine mutations?
Sorry, say again?
The familial hemiplegic migraine mutations
right.
in CAV2.1. Have you
Yeah.
thought about doing any pharmacology on those channels?
No, we haven't. We're thinking peripherally, primarily just because of the access of our compounds. I don't think we'll gain access to the CNS.
There is some of that's peripheral
Okay.
vasculature. So.
Yeah, we should look at that. Thanks for pointing that out.
Isn't that meant to be a gain of function, though? We don't know.
You could tell me.
We don't know.
I don't know what the story is.
I'm just curious, how long do you think these patients would have to take a regimen that's like this? Would it,
I think this is a symptomatic approach, so you would have to take this continuously for your entire life, the way that they currently take 3,4-diaminopyridine. And in our initial PK studies, it's very similar half-life as 3,4-diaminopyridine, which patients take every 4 hours. Take a pill every 4 hours, basically just to maintain strength.
I have one last question then. A little bit of a... Oh, Bruce, did you want to ask a question?
Just curious,
You can take mine.
in normal mice, is there a performance enhancement for grip strength?
Yeah, it's a good one. We wondered if we made a performance-enhancing drug. There's not a
Are
performance...
you looking for a bigger market? Is that right?
There's not a performance enhancement because, you're really just increasing the transmitter release magnitude, and once you're above threshold for postsynaptic muscle contraction, unless you can cause the muscle to fire 2 action potentials by really giving a massive amount of release, you're not going to really increase strength. And we don't do that, and we think that's in part because action potentials are so brief. The drug really only modifies a small subset of the calcium channels, and that's why you only get a modest increase with GV58 alone. If you do both together, there's, I suppose, the potential that you could fire 2 postsynaptic action potentials, but we haven't seen any evidence of that when we look at our ex vivo recordings from neuromuscular preparations. So as long as you're above the safety factor of postsynaptically, probably doesn't matter that you release more transmitter.
Well, thank you. I think that was great. And you're in an area where there's huge need. And so any kind of symptomatic relief is going to be really significant. So thank you.
I hope so. Thanks.
Great. Thank you, everybody. And some closing remarks, I think.